Started about in earlier time. What was there? Teachers used to be too much active, and he wanted to make the students active. But now students have to be much more active, and teacher will help in some doubts only. That is all. That's why you have to be smart. You have to be active. You have to present. Okay. And if the lightning is not sufficient, Srijal, you can increase also. Eh? Okay. So what is today's date, children? 2000, 2081.
Your time sorry, Miriam.
Jestha 13. Sunday, right? Oh. So time is running very fast. So we have to do accordingly. And I checked your unit test questions. Solution. Questions is already given to you. Solution was checked, and more or less you have done better this time. Give yourself a big round of applause. Yes. But you must understand the matter, because really tough time is approaching very soon. When you pass class 10, you will go to 11. At that time, you must focus on studies more, so that you have to make your base stronger right now onwards. That's why understand the matter and set in mind and express yourself. So we are discussing the chapter 4th, right? What is chapter 4th? Heredity, cell division. Next one, chromosome and heredity portion already started. We discussed about Mendel's laws also. And today, I have already given work to students. They have got little share. So little share makes huge share. Yes or no? So without wasting time, let me call on the stage the first presenter, Raju Kumar Shah.
Namaskar and very warm welcome to all. Today I am here to present on the topic of genetic technology.
I managed to. It can be done like that. So what do? I will split the screen into two halves. One side one student will do, next side next student. Okay. Who knows? Have you seen another pen in my office? You haven't seen. Who can bring? Ritik, have you seen? Okay, let me bring the thing. Then you can use this in two places. Eh? Okay. Excuse me. If the class becomes dirty, they have to pick it up everything. Eh? There is complaint. It is already clean. They have dropped many more things. Before going, they must clean. Otherwise, tomorrow morning only they have to just clean it. Is that clear? Remember. Okay, let him complete. Then I will split this screen into two halves, and roll number means second presenter and third presenter will explain about it. Okay.
My topic is genetic technology.
Listen, okay, listen.
Genes are present in all organisms, which transfer from one generation to another. Genetic engineering is a technology that involves selecting of genetic material to impart a specific character. Genetic engineering can be observed in human, animals, and food. Genes transfer from one generation to another. Thank you. Have a nice day.
Okay, that's great. So, actually, Raju's come. Raju, your duty just okay. His duty was to explain about this genetic technology, right? Nowadays, system or new word has come: genetic engineering. Where are the genes located? Inside the DNA or in the chromosome, and they have just manipulated. They have changed the genes and got new and new experiment and new organisms or new characters are developed, right? Designed. So some people they are very smart, smart in such a way that they are genius. They are keen. Keen means very interested in doing research. If those persons' genes are extracted and kept in other person's cell, then what will happen? The person also will be a genius or not. So they have started doing all those things, right? And what is the definition of genetic engineering? Please read what is given here. Technology that involves selection of genetic material to impart specific characteristics, and that can be seen in humans, animal, food, and medicine also, right? So now it's the turn of other two students. Please, two students, you come here. We have got system to divide this into two parts. Here is one thing, and we cannot write here. If you cannot write, then what to do? Sorry? Wallpaper came here. No, only this one, I think we can show. Second presenter is Bian Balami. Because if we change the screen also, then it looks nice, right? All right. So what is he going to explain? Just be focused and understand the matter. You can pause it again, so look sometime.
It is a genius to create organism with more advanced form. This method involves the modification of organism by adding or removing a single nitrogen base pair of a DNA. Thank you. Have a nice day.
Okay, he said here, okay, he's not pressing time, wait. It's not pressing time, just still listen. What is given here? Genetic engineering. So, what is genetic engineering? Technology where scientists, what is here? Manipulate means change. Okay, manipulate means they change genes to create organism with more advanced form. They are not happy with the same form. Advanced organism can be produced by manipulating, by changing, by taking them. Already we discussed about three types of variables, right? What are they? Independent variable.
Dependent variable and control variable.
Which variable can be manipulated? Yeah.
Change that is independent.
Independent variable can be manipulated. Here, genes is manipulated, genes is changed, or gene is taken from one cell and kept in another cell, one DNA to another DNA, right? So, in genetic engineering, modification of organism trait means character is done by adding or removing nitrogen base. So, could you remember the nitrogen bases present in our DNA? What are they? There are four types.
Four types.
Yeah, they are adenine. What is that? Adenine. Next one,
Thymine,
guanine.
thymine,
Then
thymine,
thiamine. And
cytosine.
these are the four types of nitrogen bases. They remove some bases which are providing higher quality and then get advanced organisms. Okay, that is all about the presentation of Bian. Now we turn Vivek Casey. You are the our class. Which fourth one? Which turn? Pujanas? Okay. Everybody correct?
Cloning, the creation of either embryos or babies that are genetically identical to living or dead parents is called cloning, and it involves transferring nucleus from somatic level to female egg from which the genetic material has been removed. At conclusion, a clonal embryo is produced. Genetic modification, the changes and manipulation of genes in living cells is genetic modification. It occurs in therapeutic and enhancement levels. At therapeutic, the illness of organism can be cured, and at enhancement level, it is aimed to improve beyond the average. Thank
Beyond
you.
the average. Okay. Thank you. So, who is the second presentator? Third presentator? Viveki. Viveki, she has just brought some, actually two terms. What are the two terms? One is cloning, another is genetic modification. So, Albert Einstein, he has already died. He died in 1955 AD. When did he die? 1955 AD. But if there was this technology, cloning technology, they could make another Albert Einstein also. Very easy. It doesn't matter how old the person is. Simply what is needed? Cell is needed. Cell. Right? Chromosome. What is given here? The creation of either embryos or babies that are genetically identical. Genes are same. So if Raju wants to have babies similar to Raju, then it is suitable. Cloning. Understand?
It will be same or not?
So all characteristics, everything. Because what is done? Transferring nucleus of somatic level. Somatic cell means that diploid cell. Any cell, nose cell, muscle cell, or blood cell, any cell is taken, and from that cell they take out nucleus. What is taken out? Nucleus contains chromosomes or not? How many chromosomes are there in somatic cell? 23 pairs or 46 chromosomes are there. That nucleus is taken, and that nucleus is kept in egg. Egg's nucleus is removed, and whose cloning you want to do, you want to make, it is kept over there. Then fertilization takes place, and later on baby is produced. So that is 100% identical to its parent. Genetically, no difference. Okay. Now, what is given here? Number second, genetic modification. So the changes and manipulation of genes in living cells is called. What is done here? Genes are changed. Genes, where are they found? DNA, especially nitrogen bases are changed. What are the four nitrogen bases? Adenine, thymine, cytosine, guanine. Some nitrogen bases are changed or modified. So, occurs in therapeutic and enhancement levels to increase their extra characteristics. Like the person is dwarf, but very much talented. Yes or no? But what they want to do? They want to produce person with strong physique, tall also, genius also. At that time, this method, genetic modification, can be applied. For that, which gene is responsible for growth, that is taken and kept in another cell, and process is carried out. Okay, now whose turn? Pujana's turn. Okay, Pujana, please come on the stage.
Hello.
Give me that one. Where is that?
Trait selection is a selection of sperm, eggs, or embryo that possess the. So what is this?
Genes.
Genes which are associated with certain traits. So in this process, the presence of certain genes is determined in eggs, sperms, or early embryos. The desired egg, sperms, or early embryo carrying the genes of interest are utilized to create a baby. This technique can be employed for either medical or non-medical purposes.
So for medical purpose and non-medical purpose, what can they use? This trait selection. Actually, how many methods did we discuss, hello students? Three. What is that? No, in genetic engineering, cloning, and genetics modification and trait selection. Okay. If so, now we are going to discuss here one thing. Last time, the duty was given to some students. Um, so Sonish is going to explain now the work done by whom? Luniva. Okay. Genetics technology. Yes. So ready? Okay. Now you come here. Come here. Come here.
यार बंदा सो
Okay. One by one you can do. Listen now. Give your friend introduction and continue. Yeah. This is very good system.
Good afternoon, everyone. I'm I'm Sunjin from grade 10, standing in front of you to give an explanation about genetics and genetic. Genetics and genetic technology. So the first topic is heredity. Heredity refers to the passing on of characteristics from parents to the offspring through their genes. The first organism was unicellular and very simple. However, many well-developed and complex organisms now exist distinctly on Earth. A condition that develops clear and definite difference between organisms is called variation. The branch of biology that deals with the mechanism of heredity and variation is called genetics. Gregor, John, and Mendel founded the law of genetics. Thus, he is known as the father of genetics.
Wait.
The characteristics of pea plants selected by Mendel for his experiment are all are all are all shown in this figure. In the okay.
You want to go next?
No sir. So here are the seed of the flower pod and stem. And sorry, next one. So here are the characteristics, dominant traits and recessive traits. So here are the characters as color of flower. Its dominant traits is purple, and its recessive traits is white. Another one is a position of flower. Its dominant traits is axial, and its recessive traits is terminal. And and uh, and the height of plant is dominant tree tall tree, and recessive traits is dwarf tree. And same and shape of speed and shape of seed is dominant traits is rounder, and its recessive traits is wrinkle. And it is the color of seed. Its dominant traits is yellow, and its recessive traits is green. And shape of pod. Its dominant traits is inflated eye, and its recessive traits is constructed small eye. Sorry, next one. So So why did Mendel select a pea plant for his experiment? Because a pea plant is naturally self-pollinating, and artificial cross-pollination can also be performed on it. This helps to maintain the purity of character in offspring. It has a number of distinguishable constructing characters such as a tall and dwarf pea plants, red and white flowers, round and wrinkled seeds, etc. Thus, the result of experience can be checked by observing different characters. The hybrid plants obtained as a result of cross pollination in tree plants are also fertile. In many cases, it is found that such offspring are sterile, and the life cycle is of short duration. So the results of experience can be obtained in a short period. The plants are convenient to handle, so they can be grown anywhere.
Okay,
Thank you.
so that is all about this one. Mendel's experiment and Mendel's reason for selecting right? And today, how many students they are presented? 1, 2, 3, 4 they have done, right? So, with the person having fifth diploma, yours. So, what is your duty? Prepare slide and send me. Only one slide is enough. Understood? Major points so that you can come and explain. Okay? So that by combining all those things, our class can be completed. Clear? Okay. Thank you.